Yes, ma'am. Yes, ma'am.
Morning, everybody.
Morning, sir.
Good morning, sir.
Good morning, sir.
Good morning, sir.
Good morning.
Good morning, sir.
Good morning, sir.
But you are not allowed to talk now.
When I greet, didn't you see the instruction? When you are asked to talk, then you unmute and you talk. That's what I said. Many of you will see the instruction, you will not even bother to read. I greet you. So what stops you from now responding? So back to mute mode. Mute yourself now. We are starting the class. On Monday, we were discussing some terminologies that we use in genetics, and we stopped at genotype and phenotype, which are being represented by this slide. This is the slide where we stopped. And you could see from the slide when we are talking of phenotype, which is the expression of the genotype. So you can see the two diagram slide. Hello, if you don't mute yourself, I will expel you from the class. I will not say anything concerning mute or not mute yourself again. Once I found out that you are the one talking and disturbing the class, I will just expel you. And you know the implication of that. You won't be able to come in again. Somebody would have taken your place. So mute yourself. When I ask a question and you need to respond, raise up your hand. That is the instruction I gave before I started. So now you can see from the two pictures, the diagrams of the plants, one that is tall and the one that is dwarf. So when the phenotype is tall, you see it, that's what we can see. And when it is dwarf as well. But the genotype may differentiate between all those tall plants or individuals, because some are homozygous tall and some are heterozygous tall. Homozygous tall means the two dominant alleles are present. That's the capital TT. When it is hetero, like you know, hetero means different. That is, the two alleles are not, similar in the sense that one is dominant and the other is recessive. But because in the presence of a recessive allele, dominant allele will still be expressed. That is why tallness is still being expressed even at heterozygote state. So we continue from there today. Like I said in the previous slide, homozygous, a condition in which both the members of an allelic pair in the homologous chromosome are identical. Can you see that? So they can either be dominant or recessive allele. So tallness, which is capital TT, that is the two allelic pair are identical. Okay, don't worry. Then when you talk about dwarfness, the two alleles too are identical. So it can either be dominant or recessive allele. So homozygous tall, that's capital TT. Then it can also be homozygous dwarf, that is small tt. Adedoyin, I'm giving you the last warning. You are not muted. If care is not taken, I will send you out of the class. That's the last warning I'm giving you. What is too hard in following a simple instruction? So that is homozygous. We can either be homozygous dominant or homozygous recessive, as long as the two allelic pair present there are identical. Now, when it is heterozygous, it's a condition in which the members of an allelic pair in the homologous chromosome are not identical. So we can have one dominant and one recessive allele. So the two allelic pair in the homologous chromosome are not identical. Okay, so you can see the tallness is being represented by capital T, small t. You cannot have dwarfness in heterozygous. It's not possible. Okay, because heterozygous simply means the two alleles are not identical. So how can you now have heterozygous recessive when recessiveness can only be expressed in the absence of a dominant allele? So another terminology is hemizygous. Hemizygous is a condition when a gene are present only in one copy. For instance, sex chromosomes. The sex chromosome in human are X and Y. So the genes in X chromosome in male have one X and one Y chromosome. Okay? One X and one Y chromosome. That is, a male is XY. And genes on Y chromosome is only one Y chromosome in males. In male is only one Y chromosome and only one X chromosome. So in other words, a male is said to be hemizygous. That is, containing one copy of the gene, not in two now. So like the X chromosome in a male is only one. Like the Y chromosome in male is only one. So these are hemizygous. Okay? So when the two are present, okay, so we call it when it is XX, that will be homozygous. Okay? When it is XY, that will be heterozygous. But when it is only one X, that is hemizygous. Now, dominance. Dominance is the ability of an allele to express itself phenotypically, both in homozygous, that's capital TT, and in heterozygous, that is capital T, small t. If a gene is able to express itself, whether in homozygous or in heterozygous, such an allele is said to be dominant. Then recessiveness. Recessive allele will only be expressed in the absence of a dominant allele. Recessive allele will only be expressed in the absence of a dominant allele. And that means it will only be expressed when the two alleles that are recessive come together. So the inability of an allele to manifest its phenotype in heterozygous condition is said to be recessively, because it can only be expressed in homozygous small tt. For instance, cannot be expressed in heterozygous state. So that is the the meaning of recessiveness and dominance. Uche Angel, what's the problem? You are raising up your hands.
Hello, sir. Good morning, sir.
Yes.
Sir, please, can you explain more on that Hemizygous? I don't really understand.
Hemizygous, you know, of course, you know homozygous and heterozygous, right?
Yes, sir.
You
Yes,
observe
sir.
that in homozygous, there are two alleles that are identical, like I'm showing here now. Homozygous, tall, that's capital TT, right? Heterozygous is the two alleles are still present, but only one is dominant, then the other is recessive. That is capital T, small t. Now, when it is hemizygous, is when the other is not present. That is, only T is present. For instance, let's say only capital T is present. The other capital T is absent. That is T naught. That is only one copy represent the gene instead of two copies of allele to represent a gene. Only one is present. Of course, it happens in during non-disjunction, during meiosis, which you have been taught. So when only one is present, then you call that hemizygous. For instance, have you heard about Turner syndrome before? Turner syndrome.
No, sir.
Okay, it's a disorder or a condition that affects male. Rather than the male, that affects female, sorry. Rather than the female having XX, you know, that's the normal thing for a female. The female will now have X naught. That is, the other X will be missing. So only one allele will be representing that gene. The other allele is missing. That is hemizygous. So we represent the missing allele by 0. So if you want homozygous tall here to be represented as hemizygous, it will just be T naught. That is T0. That means that other one is missing. And the only allele is now representing the gene instead of two alleles. Is that clear now? I can see many of you raising up your hand. I think we have to.
Yes, sir. Well understood.
What?
I said, yes, sir. Well understood, sir.
All right. So others that are raising up their hands, we have to delay that until towards the end of the class. Otherwise, we just be entertaining your question and we will not move. So you are noted. When we are about rounding up, I will ask for your comments or your question. So another point is hybridization and hybrid. Hybridization is the process of crossing two genetically different individuals. Two genetically different individuals. So we can say crossing between a man and a woman is hybridization because the two of them are genetically different. One is a male, one is a female. So they are genetically different. Whatever they produce is now the hybrid. Is that clear? Hybridization is the crossing of two genetically different individuals. So a male with a female, crossing the two of them is hybridization because they are genetically different. Mind you, genetically different in this case does not mean they are of different species. They are of the same species, but when you analyze their genetic composition, they are different. Of course, a woman is different from a man. Is that not so? Until nowadays that you now want the two to be the same. You want a man to be a woman and a woman to be a man. Mixing up things. causing unnecessary distraction. So a man is different from a woman. So when they mate, okay, the process of mating is hybridization, crossing them. Whatever they produce as a result of the mating, that is their children, they are the hybrid. They are the hybrid. So, for instance, now we cross a homozygous tall plant here with a homozygous dwarf plant. You know, they are genetically different. Okay, homozygous tall will produce gametes that will contain capital T, and homozygous dwarf will equally produce gametes that are, you know, contain small letter T. Okay, so it is these gametes that will meet and fertilize each other to now produce offspring, which is capital T small t. And that capital T small t is heterozygous tall. Heterozygous tall. Now, in a situation where a man that is fair in complexion, okay, marries a woman that is dark in complexion, their children complexion will show a hybrid. In other words, their children are the complexion of their children, or in terms of complexion, relative to the two parents, their children are the hybrids of them, because they will now combine the alleles in the two parents to have their own genotype. You will see that their genotype is different from either of their parents. Capital TT by small tt. And in them, you now have capital T small t, which is heterozygous tall, because capital T is dominant over small t. So they are the hybrid. You can see the progeny of hybridization is called hybrid. Hybrid. So when you find out that there's a trait in your dad, and there is another contrasting trait in the mom, and when you now look at yourself, because they must have passed those traits to you, you now see that, oh, you have a kind of a mixture between the two. Then you are an hybrid. You are an hybrid. So that is the progeny of hybridization. So then we go on to monohybrid and dihybrid. An organism which is heterozygous with respect to only one pair of allele at a locus under study. An organism which is heterozygous with respect to only one pair of allele at a locus under study. So when we have tall by dwarf, okay, you know, when you have tall and you cross it with dwarf, you are going to get tall, because tallness is dominant over dwarfism. So you are going to get the progeny that will be tall. That shows an allele, capital T, has combined with an allele that is small t. So that means at that locus, what you are crossing is only involving one character, one trait. That is mono, one. Then hybrid, that is, you want to see the inheritance of following the inheritance of a trait, So, for instance, you only take height. Height can be tall and can be dwarf. That is one trait at a time. You cross that and you want to know what the outcome will be. So crossing or following the inheritance of a trait is monohybrid. Then when it is hybrid, dihybrid. An organism which is heterozygous with respect to two pairs of alleles are two loci under study. That is, following the inheritance of two traits at the same time. We want to see when two traits are crossed, how the two traits will reflect in the offspring so produced. So here there are two traits. Yellow color. That is, you are talking of color and the shape of the seed. There are two different traits. Color and the traits. And the shape. So color, that's yellow. Shape, round. We want to know the inheritance of these two traits. When you now cross this, in most cases, you cross heterozygous of the two, heterozygous of yellow and heterozygous of round, because it has been shown by Mendel that yellow is dominant, the yellow color of seed is dominant over green color seed. And as well, round shaped seed is dominant over wrinkled shaped seed. So you now cross a seed that is yellow and round with another one that is green and wrinkled, and see what the hybrid will look like. So we talk more about this under Mendelian laws of heredity. You see how it is demonstrated in monohybrid and in dihybrid. So, okay, now we are now showing a monohybrid cross. Okay, that shows a cross between two individual organisms which differ from each other with respect to one pair of allele under study. So we have seen monohybrid before. That is looking at one trait. So when you now cross monohybrid, that means you are monitoring the inheritance of a trait, one pair of allele under study. How will that trait be, transmitted from parent to offspring? Just one trait at a go, mono. Then di, like I said, is a cross between two individual organisms which differ from each other with respect to two pairs of allele. So, of course, from the name, mono, then di. Hybrid, we have seen crossing. That is, you cross. So, in this case, you can see yellow and round seed being represented by this genotype. The yellow color, because it's dominant, is YY. The round shaped seed is represented by a capital RR. So cross with green wrinkled seed. What I said, green is recessive. That's why it is small YY. And wrinkled too is recessive. That's why we have small RR. So when they are crossed, you are going to get the dominant phenotype in the offspring. Okay, but that dominant will be heterozygous. It won't be homozygous again. So we will end up getting yellow and round seed. But their genotype will not be capital YY and capital RR because they are now hybrid. They are now hybrid. Now we talk about F1 and F2 generation. F simply means Filial. Filial meaning son. So F1 are the son, that is the first generation, which are the sons and daughters of the first generation, that is their parents. So F1 simply means the first generation progeny of a hybridization. That is the first generation from parents now. Parents giving birth to children. So their children is the first generation, which are the F1. Okay? When you go to F2, that is the children or the grandchildren of the parents, which will now be the children of the F1. So F2 are the progeny of hybrid, that is of F1, when it is hybridized with any of its parents. So you can go on and on having F1, F2, F3, F4, and so on. So for instance, suppose we cross homozygous dominant for tallness with homozygous recessive for dwarfness. The gametes. I want you to follow the crossing. In this crossing, they didn't just start drawing the line from capital TT and draw the line from small TT. The capital TT will produce gamete. It is the gamete that will be involved in reproduction and not part of, and not this individual directly. They produce gametes. The gamete produced by capital TT will all be capital T. The gametes, that is sex cell now, produced by small TT will be small t. So it is those gametes that will fertilize each other to now form F1, which are the capital T, small t, which are also the hybrid. Okay, so this is F1. Now there is a phenomenon whereby you now cross the F1 offspring with the genotype, with the recessive, genotype of the recessive individual. That is one of the parents now. You cross the F1 with the parents having recessive allele, that is by TT. We call it Backcross or test cross. Test cross. So this is the recessive parent, like I said, which we cross it with. Then each of them too will produce gametes. Look at the gametes produced by this individual. Is no longer one, because some gametes will contain capital T, some will contain small t as a result of meiosis. But because this individual has the two alleles identical, they only produce one type of gamete, which is small letter t, so which we now combine in terms. So suppose capital T combines with small t, you have this. Okay? And when these two combines in turn, we have this. But when we complete this, we have two of these, because this and this will give us this, right? But because it's only one, that's why we did not bother ourselves. It's going to be, so this one we combine with this different one in terms, and we have this, and we have that. So these are the F2, which is like F1. So you can have different combination. It depends. So then we call it so that is what is termed reciprocal cross. We have what is termed reciprocal cross. In reciprocal cross, what do? We reverse the sexes of the parents. Suppose in the first one, okay, in the first one that I taught, maybe it is the male that is taught that we cross with a dwarf female. These are the symbols representing male and female. Look at them very well. So whenever you see a symbol like this, it represents a male. And when you see a C, it represents a female. So that is the symbol. Now, initially we did tall male by dwarf female. When we now want to carry out a reciprocal cross, we now interchange their sexes. So we now have a dwarf male crossed with a tall female. That is what is called reciprocal cross. Crossing reverse crosses in which the sexes or the parents are interchanged. Okay? So if the traits are autosomal, the reciprocal cross always yield same result. Autosomal means, you know, human beings have 23 pairs of chromosomes. So out of these 23 pairs, 22 are said to be autosome. Autosome, that is, they are born on the chromosomes that are not involved in reproduction, in sex, in the formation of gametes. So they are autosomal, and they will be inherited equally, in male and female. So male and female will possess traits that are being carried by autosome. So whether you reverse their sexes or you interchange, you always get the same result because they are not sex linked. But if the traits are on sex chromosome, that is X and Y, the reciprocal cross always gives a different result. The reciprocal cross will give different results. Okay? So because the trait is on sex chromosome and they have a way of their transmission. Now, another one is backcross. Backcross. This is the cross of F1 offspring with one of its parents. I said that one before, where you have this crossing the F1 with one of the parents. Okay? But there is backcross, there is test cross. So you can cross this heterozygous tall with either of the parents. Maybe the homozygous tall parent or homozygous, short or recessive parents. So when you do that, that is called backcross. That is crossing the F1 with one of the parents. Okay? But when you now cross F1 with the recessive parent, that is this, like it happens here. This is a test cross. Test cross. Don't mix the two. Backcross means crossing F1 with any of the parents. Okay? Whereas test cross is crossing the F1 with the recessive parents. So, like we have here now, this is a test cross, which we are going to see. So, the backcross is cross of F1 offspring with one of its parents. If F1 is crossed with dominant parents, okay, so that shows you can cross it with any of the parents, whether dominant or recessive. So if F1 is crossed with dominant parents, all progenies, that is F2, will be dominant. If F1 is crossed with recessive parents, individuals with both phenotype will appear in equal proportion. So, if, for instance, what they are saying here is if we crossed capital T, small t progeny with the dominant parent, that is capital TT, that means all the progeny that will be produced will express dominant traits. They will all be tall. But if we cross F1, that is capital T, small t, with the recessive parent. Recessive parent, that's small tt. The progeny that will be produced will show both tall and dwarf individual in equal proportion. In equal proportion. So that is so if you now notice, this is what we use in test cross. We want to know whether the progeny of F1 are homozygous dominant or heterozygous dominant. So by the time you cross and you obtain these results, then you know their genotype, whether they are homozygous dominant or heterozygous, as the case may be. So the ratio of progenies produced during backcross is called backcross ratio. Like we are going to have equal proportion, that is 1:1 or 50-50 in this one, test cross. So that ratio is called backcross ratio. So you may be asked to calculate the backcross ratio. So that's how you do it. Now I said test cross. The test cross is a type of backcross. Okay, it's a type of backcross in which the F1 progeny is crossed with its double recessive parent. Like I said, double recessive parent. So this is the F1, capital T, small t. When you cross it with double recessive parent, what we are doing is a test cross. You can see it's a type of backcross, truly. Because cross is this with either of the parent. But when it is with the recessive parent, then that is test cross. Test cross is used to determine whether the individual exhibiting dominant character are homozygous or heterozygous. So to detect the genotype of F1, I've said it that we use test cross to know whether the offspring, the F1 here, is homozygous or heterozygous. So when we cross, the test cross with the recessive parent, and we got these two. Look at these two. Then we now know the genotype or the zygosity of the individual, whether it is homo or hetero. So this is a kind of a test cross. Crossing this with the recessive parent, we now have in their F2, capital T, small t, and small t. That is ratio 1:1. So when we get this ratio, it is called test cross ratio. Test cross ratio. Like we can have backcross ratio. So a test cross is a type of backcross. It's a type of backcross. So those are some of the terminologies used in genetics. Okay, though we have talked about this before. Genes are basic unit of genetic information, and they determine the inherited characters. Then the genome is the collection of genetic information in an individual. A chromosome is a storage unit of genes. In other words, genes are located on chromosome. So this is a chromosome. Then the bands or the shaded region represent the location of genes. So you have this, you have that. These are the location of genes. And locations of the genes on the chromosome are called loci. Loci, plural, locus, that is the exact position of a gene on the chromosome. So DNA is the nucleic acid, that's the deoxyribonucleic acid that contains the genetic instructions specifying the biological development of all cellular forms of life. So gene contains DNA, which are, which encoded information, and they are in a sequence, the nucleotide sequence. So I said genes are segments of the deoxyribonucleic acid, that's DNA. DNA, you can see how DNA is obtained from the word deoxyribo, then nucleic N, the acid DNA. I said it contains the code for a specific protein that functions in one or more types of cells in the body. Chromosomes are structures within cells that contain a person's genes, and chromosome contains hundreds to thousands of genes. So on a chromosome, you will have hundreds to thousands of genes. And you can imagine how many genes we have as a human being. When we have 46 chromosomes, each chromosome can contain hundreds to thousands of genes. So when a chromosome is missing or is defective or is duplicated, you can imagine the seriousness or the effect that can cause. So if it is missing, that means all the genes, which may be thousands, are missing in the individual. So, and those genes contain information. So that means those information too will be missing in anybody that inherits such a chromosome where one is missing, because they are always in pair. So if one of a pair is missing, it can cause problem. Talk less of two or a whole pair missing. Alleles, alternative form of a gene responsible for differences in phenotypic expression of a given trait. We talked about brown eyes versus green eyes. The two are just alternative expression because it's talking about the same trait, color of the eye. A gene for which at least two alleles exist is said to be polymorphic. Okay, that's according to Mendel. A gene contains two alleles, but we have instances which was not developed or which was not discovered during Mendelian time. Mendel only discovered that a gene contains two alleles and nothing more. But later on, it was found that a gene may contain three or more alleles. And in such case, we call multiple allele condition when a gene contains more than two alleles. So this just for our information. We are not going deeper into this, because this is some advanced genetics, which we come to know as we progress in our studies. So genes are contained in chromosomes, which are found in the cell nucleus. A chromosome contains hundreds to thousands of genes, said before. Every normal human, mind that word, normal human. So many humans are not normal. That's why there are conditions, there are disorders. But when somebody is not having a disorder, it's normal. That individual must contain 23 pairs of chromosome. 23 pairs of chromosome, homologous chromosome. And that gives a total of 46 chromosomes. So in a normal individual, we expect to see 46 chromosomes. So when we are now seeing 45, that's a chromosome is missing. Or we are seeing 47. That means a chromosome has been duplicated. So normal human being has that. So a trait is any gene-determined characteristic, and is often determined by more than one gene. So we have different traits. Any trait you observe in yourself, they are under the control of genes with the influence of the environment. Environment will only influence the expression, whether full expression or partial, or as the case may be. But the gene must have been inherited for one to express it, for environment to play its part. Some traits are caused by mutated genes that are inherited or that are the result of a new gene mutation. So all these are just telling us more about genes, chromosome, and traits. So Mendelian genetics. We have said that Mendel did his work on garden pea. Why did he choose garden pea out of the many varieties in his garden? Garden peas produces offspring quickly, okay? And those offspring can easily produce again. Then there are clearly contrasting characteristics in the plants. Okay, you notice that some of the plants are, in terms of height, some are tall and some are short. In terms of the seed shape, some are in terms of the texture of the seed, okay, some are smooth, while some are wrinkled. So you notice contrasting pair, even in their seed color, in the flower color. So they show contrasting characters. And that is what fascinated him to work on that. So Mendel also proposed laws, the two laws. In fact, there are three. So these are the characteristics Mendel studied on garden pea. So he studied the length of the stem, that is the height. He found out that some are tall while some are dwarf, and also tall is dominant while dwarf is recessive. So these are the 7 pairs of contrasting characters. So when we hear Mendel study 7 pairs of contrasting character, they are contrasting in the way they are expressed, but he's talking about 7 traits, 7 traits which have contrasting, which have 2 contrasting ways of being expressed. So the height, tall, dwarf, position of the flower, color of the pod, shape of the pod, shape of the seed, color of the seed coat, and the color of the cotyledon. And you notice these contrasting characters in these same traits. So this another table showing the expression of the characters. Okay, so we may not be able to appreciate the purple color because this color, this is not a colored slide, but white. Then the position of the flower, axia, terminal, seed color, yellow and green, seed shape, you can see round and wrinkled. Then the pod shape, pods, some are inflated and some are constricted. The color, some are green while some are yellow. The height of the plant, tall and dwarf. So these are the traits, the 7 traits that Mendel taught that have contrasting ways of being expressed. So these are the 2 laws of Mendel: the law of segregation and the law of independent assortment. They are the 2 most known laws of Mendel. So the third one, the law of dominance, he equally proposed it, but that one is not valid. It's not 100% valid. To an extent, the law is valid, but not 100%. So that is why it is not a law. You know the sequence of events leading to the formation of law, the scientific methods, remember. So the 2 Laws that are valid up till today are the law of segregation and the law of independent assortment. The law of segregation states that during gamete formation, the alleles for each gene segregate to separate from each other so that each gamete carries only one allele for each gene. The law of independent assortment states that genes are different traits. The genes for different traits can segregate independently during the formation of gametes. We are going to explain more on these two laws during the next class. So let's stop here for today. Anybody with question? I can see Joshua Anyawo and is still up. So unmute and ask your question one after the other. When I call you, to unmute and talk. Anyawo, I recognize talk. So when you're talking about I can't hear you, but I know you have unmuted yourself.
Okay, sir. Can you hear me, sir?
Yes, I can hear you now.
Yeah. When you're talking about hemizygous, so a condition when genes are present only in one copy. So he said it's common to male. And
I didn't say
when
it's common to me. Don't, when you want to quote, don't misquote. I'm not saying it's common to me.
okay.
When you now say male, that means you have generalized. Not, is it only human male or other males of other organisms?
Okay.
So in human
Okay.
is hemizygous for, especially male, is hemizygous for X because you only have one X in your genotype. You don't have two X. So a female is not hemizygous because a female has. So a male is hemizygous for X and hemizygous for Y. But a female has no Y at all. So you can't talk about hemizygous or whatever for a female. So a female has the two X, which is homozygous for X. A male has only one X, which is hemizygous. And you remember, I said homozygous is when the two alleles are what? Are identical.
Yes, sir.
That's XX. But when in a female, in a male, you have only one X, so there is no other type. You can't say whether identical or not. So it's hemizygous. So
yeah, I wanted to ask about the effects of that Down syndrome that is in the female. Does it mean that you were trying to say something about it, but I was trying to get it to this
yes.
I don't know what you called it, Down syndrome, or what's the effect to the
Turner
female?
syndrome. Hold on. Turner syndrome is as a result of non-disjunction during meiosis, so that instead of the two chromosomes to separate and go to different gametes, they will not. So a particular gamete will contain all the two, and another gamete will not have any one. So if the one, a missing chromosome, is fertilized, okay, by the X, maybe X sperm of the man, so that means a male will result. Why will it, a female will result. Why will it be a female? A female will result because there is no Y. Y determines maleness or femaleness. So when there is Y, then a male. When there is no Y, then a female. So because it's only one X, and the other one is null, that's the word, null, then a female will result. So there is a disorder anyway, and there are effects or symptoms and then the consequence of such. Okay, one of them, which I will not tell you everything because Google is your friend. You can always get Turner syndrome online. So that will be, the individual will be a female, but sterile won't be able to reproduce because the reproductive organ of the individual will be poorly developed, will not be developed properly, coupled with other effects which you can get online. So that's that for you. Is that clear?
Thank you, sir.
Steven, unmute and let's hear you.
Hello, sir. Good morning, sir.
Good morning.
Sir, can organism with the same phenotype possess the same genotype? That's my question.
An organism with the same
With the same phenotype possess the
yeah,
same genotype.
have the same genotype.
It's like organism with the same phenotype. Like that, can they possess the same genotype?
Organism with the same phenotype. You are comparing many organisms of the same species now, not just one. That's what
Like
you are
the same
saying?
species. Like the same species, yes, sir.
Like, for instance, now, children of the same parent, right?
Yes, sir. Yes, sir.
That have the same phenotype. Is that what you are
Yes,
saying?
yes, sir. Like, can they have the same genotype and phenotype at the same
Well,
time?
they can and they may and they may not. It depends on the combination that they inherited from their parent. Remember, if they have the same phenotype, let's say tallness. Remember,
Okay.
tallness can be homozygous, that's capital TT, and can be heterozygous, capital T small t, but you will not know because what is expressed in them is tallness. They are all tall. So,
Okay.
but
Yes.
their genotype may not be the same.
Okay. Okay. Thank you,
Right?
sir.
Yeah.
Yes, thank you.
Welcome. And then, okay. So, any other person? By God's grace, we meet again on Friday. And Princess Chidalu,
Yes,
unmute and talk.
good morning, sir. Sir, please, can you explain that monohybrid and dihybrid again?
The time is fast spent. You join late.
I didn't
So
join, so
there is nothing that is difficult about homozygous and heterozygous. Homo means
no, monohybrid and dihybrid.
okay. Of course, from the words, mono means how many?
One,
Di nko.
two.
Now, when you are following the inheritance of one trait at a time, you don't want to mix up. You
Yes,
know, there are many traits that happen that are inherited from our parents, and they happen at the same time during fertilization. That's the law of independent assortment that I will explain later. But you may not want to concern yourself about how others are inherited. You just want to follow a particular trait being transmitted from the father to the offspring. That, okay, the father is tall. I want to monitor the height of the offspring. Okay, in the offspring, two are tall, these are dwarf, and these are, you have the combination. And that's F1. Then you want the F1 to go to F2. That is those 1, 2 to reproduce. You want to monitor what happens in their F2. That is monohybrid. You are taking one trait. But when you want to take two traits and see how they are being transmitted, like, okay, the father is tall with dark complexion. There are two traits now.
okay.
There are many traits in us, which it will be so cumbersome. You want to find the inheritance of all these traits at a time. And that is why an individual is totally unique. No matter what, you are not or will never be the same with any of your parents. You can never be the same. You are unique in your own way because your genetic composition is quite different from their own, despite the fact that they were the one that gave birth to us. We are unique. Remember, during meiosis, there's what is called crossing over leading to recombinant, and so there is a kind of mixing up of genes before they gave it to us. So we are not like them again. That's why the fact that they are the one that gave birth to us. So when you consider so many traits about yourself in comparison with your parents, you will see so many areas where you are different from them. So you facially, you may look like them. Oh, I resemble my father. Oh, it's my mother. I walk the same way he walks. Oh, I have the same, shape of your face. Oh, with my father or with my mother. At times when people see children, say, oh, this child looks like his father. That is look. What about other traits? It's not only about looking. There are other things that are not seen. I told you, phenotype goes beyond physical observation. It extends to physiological. That is what you cannot see but are expressed. So that is it. And that is mono and di. That is, mono means you are following the inheritance of a trait. Di means you are following the inheritance of two. It can equally be tri. Okay, trihybrid, quadrihybrid. But when you start following that, it will become so cumbersome. So nature is so perfect that everything will just combine in its own way and give the results in the offspring. Now, if you now want to follow that many, you run into confusion. So that's why we limit it at 2. In fact, when we get to 2, you will see the possible combinations that we have. So is that clear?
Yes, sir, I understand now, sir. Thank you.
You're welcome. Afolabi now, unmute and talk.
Yes, sir.
Afolabi,
Good morning, sir.
good
Yes, sir.
morning.
Good morning, sir. Yes,
You
sir.
are?
Please, we would really need the slides, sir.
I will send the recording. You get the recording, not the slides.
Okay, sir. All right, sir. All right, sir. We appreciate
Okay.
and that of Monday too, sir. Thank you very much, sir.
I've sent that one.
Oh, really? Okay,
I
I will
sent the
ask my
recording of
colleague.
Monday. I sent it. Check
Okay, sir.
the platform. I've sent it.
All right, sir. All right, sir. Thank you, sir.
Okay. All right.
Yes, sir.
Aisha,
Yes, I wanted to ask. I don't exactly
I
understand
know
the
many. Do you
difference
have
of.
another class between 9 and 10?
I think they have
Yes,
chemistry,
sir.
sir.
Yes,
Those
a
that
class
have,
is
I think they
currently
have
on
left too.
chemistry.
Those that have left. It's your question that is keeping me waiting. So, no problem. Go ahead.
Yes, sir. I think that I don't exactly understand the real essence of the backcross. Why you are the F1 the backcross with the recessive parent or the dominant parent.
The Backcross.
Yes, sir.
You don't really know the essence, Abi?
Yes, sir.
Okay. And I told you the essence. When you do a Backcross, in other words, a Backcross simply means crossing the F1 with the parents. You can be with the parent with homozygous dominant, or with the parent that is heterozygous dominant. Okay? You may be asked to do that so that whatever you obtain, you can state it. So backcross, as explained, is just for that. But remember, test cross is a type of backcross. Okay, so that is why backcross comes in. Your backcross involves crossing the F1 with either of the parents. So in which, when you now do that with the recessive parent, then you are doing the test cross. So you may want to know that these our F1, they are all tall. If they are tall, how will you now be sure that their genotypes are homozygous tall or heterozygous tall? Or how many are heterozygous, how many are homozygous? That is when you now have to do the, it's now a test cross. You want to know. When you carry out a test, you will get observation that will inform your results or your decision. That, oh, having done this with this, I got this. Therefore, this is that. So you will be able to tell the zygosity of the F1, whether they are homozygous or heterozygous, but you won't know because what we'll be showing in them will be the dominant traits. The phenotypically, you will see that they are all tall, for instance, but you may not know whether that tallness is homozygous or heterozygous. So, but when you now do the test cross and you obtain, as I have said, if all of them are tall, all the offspring obtained as a result of test cross are all tall, that means the offspring that you cross with the parent recessive is homozygous. But when you now get half tall, half dwarf, so it shows the F1 you cross with the recessive parent is heterozygous. So you are able to make your decision now. So that is the essence of backcross and by extension test cross. Right? So no more question. So ladies and gentlemen, we'll see you again next class. Bye for now and enjoy the rest of your day.